So we have seen what forces define a protein structure, but in between all these myriad of forces, can a regular structure appear? And that's our topic today. Secondary structure. Somehow, proteins like to fold in two or three secondary structures. Now, let's look at them. So, if we look at the hierarchical pattern, primary structure is pretty much the sequence of amino acids, while the secondary structure is local folding of residues into regular patterns. And one thing you need to take into account here is that secondary structure doesn't it doesn't include the details of the fine packing of amino acid side chains. And a lot of secondary structure combine together, they come together to make a tertiary structure. And one tertiary structure, ultimately, if a protein has many subunits, make a quaternary structure. Now, the discovery of the first regular structure came from one of the giants himself, known as Linus Pauling. He is the only person in the history to win two unshared Nobel Prizes. One of the other person who won two Nobel Prizes was Frederick Sanger, but he had shared Nobel Prizes. But when it comes to Linus Pauling, he won Nobel Prize without the contribution of any other scientist. He's known as father of quantum chemistry and molecular biology. So he proposed the alpha helix, which is one of the foremost secondary structure, and he also proposed the beta sheet structure with Corey. So some of you might have seen this very famous show on the television. It's a sitcom known as Big Bang Theory. And I have a question. Do you know in which university it has been shot? And all the characters are from which university? They are from California Institute of Technology. And Linus Pauling also did his PhD from there. One of my very good friends, he loved Linus Pauling. And if any one of you has this problem that their chemistry is not good, they should go on Amazon or go to a bookstore and buy his textbook on chemistry. It's He wrote a wonderful textbook, and it will just make your concepts crystal clear. It's a bit outdated, but definitely still worth a read. So, his prize motivation was for his research into the nature of the chemical bond and its application to the elucidation of the structure of complex substances. He also single-handedly discovered the molecular basis of the disease sickle cell anemia, which we have discussed before as well. Let's see that how ultimately we reach this conclusion that there are ordered arrays of atoms and ultimately there is a regular folding. One of Pakistani girl, she is now the youngest Nobel laureate, Malala Yousafzai. Before that, it was this guy, William Bragg, who was the youngest Nobel laureate. And at the age of 25, he gave this idea that it is possible to calculate the position of atoms within a crystal by using an X-ray beam. And then Max Perutz and John Kendrew, who were both at Cambridge, they ultimately proposed the structure of hemoglobin and myoglobin. So hemoglobin is the protein which carries oxygen in your blood, while myoglobin is a protein which stores and carries oxygen in your muscles. But we cannot ignore the contribution by an Oxford scientist, Dorothy Hodgkin, who basically made it very clear that proteins are not colloids of random structure. Prior to this, people used to believe कि कोई inert carrier होगा जिसके ऊपर कुछ functional moieties लगी होंगी. They were not thinking कि it will be long polypeptide chains जिसकी वजह से ultimately protein एक function ultimately एक assemble करती है और एक functional और उसमें एक functionality आ जाती है. And basically what she did was she showed that crystal of the protein pepsin जिससे उसने पहला X-ray diffraction pattern लिया. And ultimately she was able to establish कि प्रोटीन इज अ पॉलीमर, इट्स नॉट अ रैंडम कोलाइड। पूरा का पूरा जो प्रोटीन का स्ट्रक्चर है वो अमाइनो एसिड्स के ऊपर ही बना हुआ है, जो क्रिस्टलाइज हो सकते हैं, जिससे हम एक्सरे डिफ्रैक्शन पैटर्न ले सकते हैं और उससे एक रेगुलर स्ट्रक्चर इमर्ज हो सकता है। इसकी क्या इंप्लीकेशन है? इसकी सबसे बड़ी इंप्लीकेशन ये है कि अगर रेगुलर स्ट्रक्चर इमर्ज हो सकता है तो इसका मतलब है कि वेरियस प्रोटीन्स जो इतने ज्यादा फंक्शंस कर रही हैं, क्या हम उनके यूनिट्स इंटरचेंज करके कोई नई प्रोटीन बना सकते हैं? दैट्स द क्वेश्चन।